Today we're going to continue our discussion of molecular genetics. We're going to look at mutation and also at the control of gene expression, determining which genes function and which genes don't function in our genome. So let's begin with our discussion of mutation. Mutation is a spontaneous change in the nucleotide sequence of DNA. And we generally classify these in two general areas. There are heritable mutations, and heritable mutations are those that are passed to the next generation. So heritable mutations occur within our sex organs and would be passed on in either sperm or ova. And then somatic mutations are mutations which occur in our body cells. And again, sometimes these can be deadly. For example, there are cancerous mutations that can lead to cancer. That may kill you, but at least that's something that isn't passed to the next generation. Now, as we look at mutation then, let's go ahead and go back to the central dogma, kind of look at the downstream effects of mutation, and we've talked about this before, so that, again, if we do have random changes in the nucleotide sequence of DNA, to get my arrow up here, that's going to lead to transcription of RNA. RNA then will be translated into proteins, and these proteins either make up the structure of the cell, do the work of the cell, but some of them feed back on both RNA and DNA to regulate gene expression. So again, there can be severe effects of mutation as we look downstream from the DNA. All right, so as we look at causes of mutation then, some mutation is caused by direct damage to the DNA strand, and some of the things that can damage the DNA strand include radiation as well as chemicals, and we'll talk about these in a second. Also, what are known as polymerase misreads, and again, we'll talk about that in a little more detail, as well as mistakes in repair to DNA, so that when DNA is damaged, there are enzymes that can repair that damage, but sometimes mistakes are made there. So let's begin with DNA polymerase misreads, because this is actually a significant cause of mutation. So you may recall that DNA polymerase is an enzyme that makes complementary strands of DNA, and this would be part of cell division. So chromosomes are replicated, and then cells divide. So you may recall that the DNA polymerase works from 5 to 3, and so it will read an existing strand and then build a complementary strand from 5 to 3. And so we're pretty comfortable now with the association of adenine and thymine and guanine and cytosine, so it will pair up the appropriate nucleotides and then build a complementary strand. Now, if there is some kind of, and there can be just literally spontaneous mistakes that occur there sometimes. And of course, we have our hydrogen bonds holding the two strands together. Now, certainly if there's damage to a strand, that also can confuse the DNA polymerase. So in other words, if there's something that's modified a strand in a certain way, then it may not know what to do with that strand, what nucleotide to put opposite that mistake, and that then becomes a permanent mistake in the cell. So polymerase misreads are a big deal because, again, if you have damage or alteration of one strand, every time you make a complement of that, that gets passed on to the next cell or potentially to the next generation. Okay, now consequences of misreads or misrepairs. One would be a nucleotide substitution. So the polymerase may substitute an incorrect nucleotide for the one that should be there. There may be nucleotide additions, there may be nucleotide deletions. So let's go ahead and look at some of these types of mutations. A point mutation is a mutation that occurs to a single nucleotide. It's a single nucleotide alteration. And these point mutations are of various types. There's missense mutations, which we will discuss in a moment, silent mutations, frameshift mutations, and nonsense mutations. So let's go ahead and look at some examples of point mutations and the consequences then of point mutations. Okay, so if we start with our DNA strand, and here I'm showing you, both strands. Remember that one strand will be coding for a protein and one will not, so I'm going to go ahead and eliminate that strand. And again, if we kind of look at the downstream effects here, if we've got a cytosine, that would be transcribed into a guanine. And if we look at this codon here, then that would be translated into a valine. So there should be a valine at this position in this small polypeptide. Now, let's go ahead then and consider what would happen if there was some kind of a change to the strand so that guanine was substituted for that. And that might have been the consequence of a misread or something like that. That then would mean that when this was transcribed, we would have cytosine at this position. So instead of GUA, we've got CUA. And so the question is, will we still end up with valine in this position in the protein? So if we look at that then, if we go to C and U and A, what we find is that actually that would be coding for leucine. So a missense mutation then substitutes one nucleotide for another, and that translates to a change in the amino acid sequence, a single amino acid change in our protein sequence. And again, downstream effects of that is that changing the amino acid sequence can potentially change the shape of the protein, potentially change the function of the protein, and then nature decides the value of that change. So I think one of the ideas that I'll come back to, and it's important to understand about mutation, is that mutation then can be acted upon by nature, and this is how new traits are developed. This is how new proteins are generated in nature, and then nature decides the value of this new protein. Maybe it works better, maybe it works worse, maybe it has a completely new function. Okay, so missense mutation then leads to a change in a single amino acid in a nucleotide chain. Now let's look at a silent mutation. So again, I'm going to take a different position. We're going to look at the same codon, but let's look at the thymine here, which would normally be translated or transcribed into an adenine, translated into valine again. And if we look at a change there, let's say that instead of a thymine, a cytosine was accidentally put in that spot. Again, that would transcribe into a guanine. And then translated here, our codon now is GUG instead of our previous. And let's see if that ends up being valine still. So the question is, will it still be valine in that position? And so as we look at this, indeed, in that third position, the mutation there, we will still have valine in that position. So that did not change the amino acid sequence. So when you have a change in the nucleotide sequence that does not change the amino acid sequence, we refer to that as a silent mutation. There is a mutation in the DNA, but it didn't change the amino acid sequence, won't change the shape of the protein, won't change the function of the protein. So it's silent. Okay, so again, downstream effect, no change. All right, now frame shift mutation is one that is very, very significant. And this can be caused by either a deletion or an addition in a nucleotide. So again, if we kind of start with our initial strand here, if we look at cytosine, that's one we're going to look at. We're going to look at the effects on this codon and our downstream effects. We should have valine here, followed by arginine, tyrosine, serine, and so on. So what would happen then, and again, this would be the appropriate codon. So here's our codons on the mRNA strand, and we could translate that into our amino acid sequence. And what would happen if that was knocked out? So perhaps radiation or a chemical has actually knocked that out. Well, what would happen then is if we eliminated that, that would shift the whole frame of our codons, the so-called reading frame of our codons. And I'm going to go back and forth here a couple of times. So again, if we go back previously, this should be GUA, CGG, and so on. But if we eliminate this nucleotide, that's going to shift all of the nucleotides to the left. And so as we read 3 in succession, that's going to change every codon from that point. So again, we're identifying that this one's going to be eliminated, and then everything gets shifted down. So as you can see, we're not affecting just a single codon in a frame shift mutation. Every codon from that point on will also be affected. And as we go ahead and translate that, we would not only have a single amino acid chain, in other words, change, we would not only have tyrosine in the place of valine, but every other amino acid down the line would also be affected because every codon is affected. So again, the concept behind a frame shift mutation is that it shifts all of your codons one direction or the other, depending on whether a nucleotide has been eliminated, or again, and sometimes in misreads, an additional nucleotide is sometimes inserted into a gene, and that would have the same problem. That would also be a catastrophic effect. So frame shift mutations generate completely novel proteins, proteins that have never existed before, and again, may not have any function at all, but who knows, maybe they do have a function, and then nature decides the value of that function. So again, even though we're talking about mutation, we're really looking at this overall process of evolution. There I go, I've put in the E word again, but yeah, this is a major force in evolution. New traits are generated by spontaneous change in nucleotide sequences, and then nature decides the value of that. That's what natural selection is. Those genes accumulate in a population over time. That leads to genetic change in a population over time. That's all evolution is. All right. Now, last but not least, a nonsense mutation. A nonsense mutation would be a change in the nucleotide sequence that inadvertently leads to insertion of a stop codon. So again, by changing one nucleotide, you potentially could generate a stop codon in a place where you shouldn't have a stop codon. So again, if that were to happen early in a sequence that leads to a protein, that would just basically destroy that protein. It wouldn't be generated. So a nonsense mutation then is a mutation that leads to insertion of a stop codon in an inappropriate place in an mRNA. All right, now as we look at other causes of mutation then, radiation damage to strands is a significant problem. Again, that can lead to misreads and then downstream problems. So, let's say that radiation has, somehow knocks out a nucleotide from a DNA strand. Again, we do have enzymes that will repair the strand. And of course, the great thing about DNA is that as long as you have one strand existing, you can build a complementary strand from that. So again, if we have damage to one strand, adenine is here. Of course, the thymine should go opposite that. But again, occasionally those repair enzymes will identify that, and occasionally make a mistake. Sometimes they'll put the inappropriate nucleotide in there. And then, of course, every time that strand is copied, that mistake will be carried on to the next cell and the next cell and the next cell. So again, that's rare, but it happens. Mistakes occur. Now, another kind of radiation-induced problem involves what are known as dimers. UV radiation can lead to these dimers. Particularly, it involves thymine most of the time. But what it can cause is that adjacent thymines will actually form covalent bonds between them that will disrupt the hydrogen bonds with the complementary strand. And it puts a little bump in the strand, in the DNA double helix. And again, there are so-called excision enzymes and then repair enzymes that will come along and potentially repair that damage. But also, in the case of polymerase misreads, if you have a thymine dimer here, and then polymerase is trying to build a complementary strand, it may not know what it's supposed to put here. It may not recognize that it's thymine and should put adenine there, and thymine and put adenine there. So again, it may insert the inappropriate nucleotides, it may skip nucleotides, it may put in additional nucleotides as a result of that. So this is, again, the problem of polymerase misreads. If there's damage to one strand, it can lead to misreads in the other strand. Now, additionally, as I said, there is also repair of the strand that can occur. So enzymes will recognize that little bump, take it out, and then hopefully, faithfully repair that. But again, those repair enzymes sometimes make mistakes in doing that job. Additionally, chemicals can lead primarily to polymerase misreads. And so one category of chemicals that we consider a mutagen is what are known as intercalating agents. And so again, as we look at our nucleotides here, you can see we've got nucleotide, and there's a chemical wedged in between those. So an intercalating agent is one that wedges between adjacent nucleotides. And then again, as DNA polymerase is moving along here, it's not sure what it's supposed to put opposite that. It causes misreads. It can't read appropriately. So again, it might be a base substitution. It might lead to a base addition or base deletion. Still another kind of problem is that a chemical might bind to one of the nucleotides. And this is what's known as a base modifier. It may bond to a nitrogenous base, for example. And again, as DNA polymerase moves along here, it doesn't recognize that this is perhaps a thymine, and so then it puts the inappropriate base opposite that. And then last but not least, the enzymes that actually make the nucleotides may actually bind a chemical that looks like the appropriate nitrogenous base, but isn't the appropriate nitrogenous base. And this is what's known as a base analog. So again, something that maybe looks like guanine, but isn't guanine, will be built into the nucleotide. And again, the downstream effect is this leads to polymerase misreads. Polymerase is moving along here and says, boy, that doesn't look like adenine, it doesn't look like guanine, I don't know what it is, and so we'll just arbitrarily put in a base opposite that. So again, these damages to one strand then leads to polymerase misreads, and then once you have that incorporated in the DNA, every time the chromosome's copied, that ends up being passed on to the next cell. Now, we can also have damage at the chromosomal level, and I'm going to talk about this more when we discuss mitosis and meiosis. But one example of this is what's known as transposition. There are some genes that we call transposons, so-called jumping genes, that produce enzymes that cut them out of one chromosome and insert them into another chromosome. So transposon then is a section of a chromosome that will excise itself, cut itself out, and then insert itself into a different chromosome. And of course, if it inserts itself into the middle of a protein coding gene, that could be quite problematic. So anyway, so these are pretty unusual, but again, they do sometimes lead to problems in our genome. Now, the human genome is massive, and I've mentioned this a little bit, just to give you a sense of that. Again, there are about 3 billion base pairs of nucleotides in our 46 chromosomes in each cell. And whereas we used to think that there might be 1 million genes, like when I was a kid, they used to say, oh, there are 1 million genes. And then they pared that down to about 100,000. And so now they think there's around 20,000, maybe fewer than that, protein coding genes. And when we look at those 3 billion base pairs, only about 1.5% of our genome codes for proteins. Yet about 90% of the genome is transcribed, although that number is under discussion right now. But a large amount of our genome is transcribed, yet only a small part of it actually translates into proteins. And every cell then will have the same genes. So what we need to look at then is, well, what makes one type of cell different from another type of cell? Because again, when you look at our body, we have many different types of cells. We have nerve cells, skin cells, muscle cells, brain cells, and so on and so on. And so what are the changes? If every cell has the same genetic material, how can cells be different? And what it boils down to is that some genes are functioning and some genes are not functioning. And what controls the differences between cells are which proteins are being generated and the amounts of proteins that are being generated, and also when certain proteins are being generated. So when a gene is functioning, we call that expression. So a functioning gene is being expressed. A gene that is not functioning is being repressed. And so it's important to understand gene expression and repression. So let's go ahead and look at some of the regulation that goes on in DNA. And again, we go back to our central dogma, and we're going to insert even one more little level of discussion here. So again, DNA is transcribed into RNAs. RNAs are translated into proteins. Proteins are structure of the cell, work of the cell. And then they also regulate functioning of RNAs and of DNA. And also the RNAs themselves help regulate whether they're translated, and in some cases transcribed. So there are several levels at which expression and functioning of genes can be affected. It can be affected at the transcription level, it can be affected at the translation level, and so we're going to look at some of these as well. Okay, so gene expression and suppression. Let's go ahead and look at some of the factors that activate and inhibit or suppress expression of genes. Now, one level will be what's known as chromosomal hypercoiling or packing. And so tightly coiled DNA does not get transcribed because RNA polymerase just can't get in there. So let's go ahead and look at an animation that'll give us a sense of this. Okay, so then looking at this effect of hypercoiling or hyperpacking on gene expression then, there are times when chromosomes are tightly packed or regions of chromosomes are tightly packed. And so they'll be, again, tightly coiled. These loops then are supercoiled. And the supercoils then, if we look at the structure here, you may recall that a nucleosome is a complex of proteins, histone proteins, with DNA wrapped around that, the DNA double nucleus wrapped around that. And so, again, when it's really compact like that, transcription cannot occur because literally RNA polymerase can't get access to the promoter region of the DNA. So just physically cannot get in there because RNA polymerase, along with the other proteins necessary here, are pretty large. So again, if we uncoil that or unpack the DNA, then what that does is make the strand, DNA double helix, accessible. Polymerase can bind to the promoter region and transcription occurs. So if a gene is going to function, it can't function unless you transcribe it at first. Now, it doesn't guarantee that it's going to function, but at least that's the first step. All right. So this hypercoiling or packing then, what are some factors then that can affect whether the DNA will be hyperpacked or hypercoiled or whether it will be loosened so that the promoter region is accessible to RNA polymerase? Well, some of the things that can affect this are enzymes that add acetyl groups to histone proteins. So acetylation of histone proteins, you may recall that an acetyl group is basically a 2-carbon unit. So if you add that to histone proteins, that will loosen the DNA. Also, if you phosphorylate some of the histone proteins, that will also loosen up the DNA. If you methylate histone proteins, that will hypercoil the DNA. And if you methylate nucleotides, that will also hypercoil the DNA. So acetylation and phosphorylation then of histone proteins will hypercoil, and methylation of histones or methylation of certain nucleotides in the DNA will hypercoil. So again, this will help determine whether promoter regions are accessible or inaccessible to the polymerase, and determine whether a gene potentially functions or does not function. Okay, now, in addition to that, there are also proteins that help activate genes, so-called activator proteins, and then also so-called inhibitory or repressor proteins. So again, what these do is they actually work at the promoter region to either help polymerase bind to the promoter or prevent or repress RNA polymerase from binding to the promoter region of a gene. So let's go ahead and look at an animation of that. And so what we're looking at here is RNA polymerase and a promoter. And you may recall again that polymerase has to bind to the promoter first before it can transcribe a section of DNA into RNA. And so RNA polymerase is actually going to work with other proteins known as transcription factors that help it bind to the promoter region. Now, activators are proteins that will assist the transcription factors in helping RNA polymerase bind to the promoter region. So again, if RNA polymerase cannot bind, there can be no transcription. And if there is no transcription, there can be no expression of the gene. So again, all of those will work together. So activators will work together to make the promoter accessible to the RNA polymerase, and then it can lock in and go ahead and transcribe the mRNA strand. Now I want to back that up just a little bit to show us the promoter before it's bound. Now what inhibitory proteins would do is that they would bind to the promoter region, so they would sit on the promoter region and prevent the polymerase from binding to the promoter. So again, activators assist the polymerase in binding the promoter. Inhibitory proteins or repressor proteins will prevent the polymerase from binding to the promoter region. Okay, so what are other factors that can affect control of gene expression? Well, some of these are RNAs and so-called interfering RNAs. And there are some different categories here, but a couple that are very similar in how they function, although they're derived in slightly different ways, are so-called siRNAs and miRNAs. And so the siRNAs and microRNAs work with proteins to form so-called risk complexes that will sit on the mRNA to prevent translation. And so they can do one or two things. They can either cause cleavage or breaking up of the mRNA, or they'll block bonding of the ribosomes to the mRNA so that you don't get translation. So these so-called silencing or small interfering RNAs and the microRNAs work with proteins then to prevent translation. So transcription may occur, but just because transcription occurs doesn't mean that you necessarily get expression of the gene if you can't translate it. So again, if we look at an example of this, and this is a little more detailed than what we need to know, but it basically will start off with a complex of RNA. A section of this RNA then will be spliced out and will associate with a protein, and then that RNA subunit will be complementary to some part of an mRNA. And so when this RISC complex then sits on an mRNA, ribosomes can't bind and translate, or it may also, some of these RISC complexes actually will cut up the mRNA so that it doesn't function. So again, when we look at levels of control, you can control expression at the transcription level, preventing transcription, but you can also prevent expression by altering translation, and that's where these microRNAs and siRNAs work with these RISC complexes to prevent the translation. Now, additionally, there are what are known as antisense RNAs, and antisense RNAs are small segments of RNA that are complementary to some segment of an mRNA. So again, if we have an mRNA that's coding for a protein, an antisense RNA would be complementary to that. And again, if it was in the cytoplasm of the cell, it could bond then to the complementary region of the mRNA, forming what's known as a double-stranded mRNA. And so now, if a ribosome tries to bind to this and translate, it's prevented from doing that. So again, an antisense, in a way, is similar to the siRNAs and microRNAs. It's just that they do not work with other proteins to form these RISC complexes. But again, they also prevent translation by binding to an mRNA. Now, if you ever read about RNA too, you'll sometimes see dsRNA or ssRNA, and all that means is dsRNA just means double-stranded RNA. So in the example we looked at where the antisense bound to the mRNA, that would be a double-stranded RNA. And ssRNA just means single-stranded RNA. All right. Now, other levels of control would be at the level of mRNA processing. So there are modifications that occur in mRNA. And again, we can look at an animation of this as well. So that when we look at, and you may recall with mRNA, there will be a cap and a tail added, and then introns removed, and that can generate one protein. But it can also be processed in different ways such that different introns are removed. So we can actually get two different proteins depending on which introns are removed and which exons are spliced together. Also, if you had any problems in generating the cap or the tail, that could also affect control of expression as well. So again, there are genes that are involved in controlling the mRNA processing. If you had mutations or alterations in those genes, that could also then affect expression. Other levels of control, mRNA is degraded at some point. It will be broken apart. And so the life of an mRNA determines how much protein is generated. That also determines the characteristics of a particular cell. Additionally, protein activation. There are modifications that occur to proteins. So when a protein is made, sometimes it will be split apart, or you will have additional molecules added or removed from the protein. And so again, if those enzymes are altered in some way, that will alter the overall protein at the end. So even at the end of translation, there are still levels of control by modifying the proteins. And then also the life of a protein can be altered as well. So protein degradation is also a factor here as well. So again, even at the end of the line, once the protein is made, there are still levels of control to ultimately determine whether this gene will actually function or not. So it's these changes then in control of expression, or differences in control of expression, which determine the differences in our many cells.